just to begin, what initially seemed like a novel experience for some of us, so skipping the commute to the office and working from home, has evolved into weeks and months locked away in quarantine. And as each of us search for updates, news, and signs of hope, we become familiar with terms that were once reserved for epidemiologists and principal investigators. Concepts like bending the curve, serological testing, structural therapeutic design, and RNA vaccines. If the strategy of the Weizmann Institute since its founding was providing the scientists with the freedom, infrastructure, and creative outlets to tackle the most pressing scientific questions, the current crisis has really unleashed a flood of inspired, promising, and purposeful scientific collaboration in response to the heightened sense of urgency around this novel virus. In record time, we've watched the Institute do what it does best: collaborate, share, adapt, and doggedly pursue knowledge that will eventually vanquish this pathogen. And to describe some of this in more detail, we are joined by two scientists today from the Weizmann Institute, who can detail for us some of these collaborations and the national and international leadership that the Weizmann Institute is playing right now, every day. Our main speaker will be Professor Sarel Fleishman, the Professor of Biomolecular Sciences, and our other speaker will be the Vice President of the Institute, Professor Roei Ozeri, who's helping to lead and navigate the quickly evolving situation in Rehovot. Professor Ozeri was born in Israel. He got his Bachelor's of Science degree in physics from the Hebrew University, and he received his master's and his PhD degree in physics from the Weizmann Institute of Science. He conducted his postdoctoral studies at the National Institute of Standards and Technology in Boulder, Colorado, in the group of David Wineland, who was the 2012 Physics Nobel Laureate, and he joined the Weizmann Institute faculty in 2007. I'll let Roei make some comments now.
Thank you very much, Dave, and thank you all for joining. And I'd like to use this opportunity to thank all of you for being longtime supporters and friends of the Weizmann Institute of Science. I think for us, we're always full of gratitude for our friends and supporters, and especially times like this, it means a lot to us. The situation, I don't have to tell you too much about the global situation. It's not just of course, not just an Israeli episode, but all of us all around the world are under this global pandemic. We watch from here, we know that the situation in the East Coast of the US is dire, and we're very concerned, of course. For us as well, that's the fifth of the 6th week of almost complete lockdown under the regulations that were issued by the Israeli Ministry of Health and the government, which means that for us as an academic institution, we had to lower the presence on campus to 15%. We had to send almost 70% of employees, not the students and the scientists, but the technical and administrative staff, on paid leave, which means that the activity at the Weizmann Institute, on the one hand, had to be reduced to a bare minimum. And the reasons for that are reasons of public health. In fact, very early on, so beginning of March, we had to cancel on the call all the conferences on campus for obvious reasons. But now we're starting renewing conferences through digital means, through video conferencing. And the first conference that we're holding is a conference of those 40, 50 groups that are doing coronavirus research in order for people to exchange ideas and collaborate. Something else that happened, due to the fact that science is now at the forefront of this global fight against the pandemic, that turned the media projector onto the Weizmann Institute. I think we're not, we haven't been an institute that seeks media attention too much. In recent weeks, we've been clearly at the center of both national and international media, news stories at the Israeli TV broadcasting stations, as well as all the major newspapers. Stories about research and tests performed at the Weizmann Institute are constantly showing up. And there were recent stories in the New York Times and the Wall Street Journal about research studies that were performed at the Weizmann Institute as well. Luckily, the situation in Israel seems to be slowly stabilizing. The number of new cases every day are gradually going down. I think today we're roughly at the 200 new cases per day. And this weekend, the government has decided to slowly reduce the means of lockdown, which means that now we're allowed 30% of our employees on campus. So there's gradual increase, which allows us to open up facilities that up to now were closed, such as the biological services, the chemistry facilities, all the support facilities of science on campus. And we hope that this would really help us slowly go back into full research capacity. And we're all hopeful that the coronavirus situation will slowly slow down and allow us to get back, maybe not to the full routine that we had a couple of months ago, but to a form of routine that will help us function again, both at the personal level as well as scientifically. Hope all of us share. So with that, I would like to introduce one of our front fighters against the coronavirus epidemic. And you know, we're very proud at the Weizmann Institute of being an interdisciplinary research institute. We're very proud of the fact that our science is not categorized into hard-walled categories such as physics or math or biology or chemistry, and that our scientists are versed in multidisciplines of science. And I think Sarel is a great example of that. Sarel's research is at the interface of computer science, molecular biology, biophysics, biochemistry. Sarel did his bachelor's and his graduate school at the University of Tel Aviv University. He did a postdoc at the University of Washington in Seattle, and since 2011 is a faculty member at the Weizmann Institute of Science. Sarel is an expert about taking proteins and stabilizing them, and the path Sarel takes, and I'm sure he'll tell you about it in his talk, starts in silicon chips. So Sarel uses develops the computer programs that design these proteins, and then he fabricates these proteins and tests them in the lab. And these proteins then can have medicinal capabilities. So for example, one of the big successes of Sarel in recent years was to design, test, and prove that a particular protein is stable enough to serve as an excellent candidate for being a vaccine against malaria. Sarel is now turning his innovative powers into trying and design a vaccine that would work against the coronavirus as well. I want to mention that Sarel Fleishman is also the director of the Dr. Barry Sherman Institute for Medicinal Chemistry. We have high hopes for Sarel as a fighter against the coronavirus. So please, Sarel.
Thank you all for joining. These are indeed very, very challenging times. So my lab, as we mentioned, works on, develops computer programs to design better proteins. And we have a specific focus on antibodies, which are special molecules that our body produces to fight off viral infections. In fact, when our body first encounters a new virus, it studies the virus surface and tests a very wide array of different antibodies that the body produces in order to test which one of them can bind and block the virus infection. Once it finds such an antibody, it mass produces it to fight off this particular infection. And it also stores this antibody in what's known as immune memory, so that the next time we're faced with this specific virus, the body can, again, mass produce this antibody, and we may not even get sick. So this is how our bodies fight off viruses. The key issue about this new coronavirus is that it is new. We haven't encountered it, and therefore our bodies do not have immune memory for this particular virus. And that means that the whole process of studying the surface and finding the correct antibody needs to take place, and that can take weeks. And that's why the disease can become so severe. So there are very many different directions that science and technology are taking in order to fight off this pandemic. And I'll just mention three, just very, very briefly, that are relevant to this talk. One of them is obviously to develop a vaccine. This is a very long-term effort. It may take as long as, some people think maybe half a year, but it may take as long as a year and a half to develop an effective vaccine for clinical use. Obviously, once we have such a vaccine, we'll inoculate the entire population and therefore provide protective immunity. But not all people respond well to a vaccine. Some people don't develop neutralizing antibodies when vaccinated, and therefore we must have another form of treatment, essentially a drug to fight off this infection. And therefore there are two other different directions that research is now taking. One is to isolate antibodies from convalescent patients of coronavirus, isolate these antibodies, find out which one of them is neutralizing against this infection, and then mass produce it and use it as a drug to treat the infection. This is ongoing and I think is very promising. And the third direction is the direction that we're most focusing on, is to develop completely new antibodies that would find vulnerabilities on the surface of the virus and block infection. So my lab is trying to help in all of these three efforts, and again with a focus on the third of designing completely new antibodies that would block the coronavirus. So now I'll share my screen with you. So I named this brief talk a progress report because this is very much what I'll present today on the coronavirus is very much quite preliminary. We don't have conclusive results yet, and I'd like to stress this at the start. We have ideas which we're very hopeful about, and we're really working very hard to realize them. So I guess many of you have seen these very haunting, images of the coronavirus. This is the coronavirus image here, and you can see that the coronavirus is sort of circular and there are these protrusions on its surface. These protrusions are known as the spike protein. They're actually viral proteins on the surface of the virus. And they're the spike protein is actually the main villain in the story of the coronavirus infection, because the spike protein enables the virus first to attach to cells that line our lungs and then to insert into these cells. So of course, by blocking this protein, the spike protein, we may be able to block infection. But these images are far too coarse, they're low resolution, and do not provide us with enough detail to design new antibodies to target the spike protein. 2 months ago, our collaborator at the University of Texas determined the atomic structure of this spike protein, and you can see it here. So I mentioned that the coronavirus is new to us, but it's not completely new. In 2003, you may remember there was the Sars epidemic, and the Sars epidemic was caused by another coronavirus, not the coronavirus that currently is prevalent, but a different coronavirus. And these viruses are quite similar to one another. And what this means is that we don't need to study the new coronavirus from scratch. We can actually transfer the knowledge that was gathered over 15 years of excellent research on the SARS epidemic and learn from that some of the vulnerabilities of this new coronavirus. And that research really very clearly outlines that there are vulnerabilities on the spike protein, and I labeled them here in yellow and pink. We know that attaching to these sites can block the spike protein from infecting our cells. So if we could design proteins that would bind to these specific sites, we could block infection. These could serve as potential therapeutics. So the key question becomes, can we design proteins that don't bind just anywhere on this large spike protein, but bind specifically to these very sites? So this is taken from work that I did as a postdoc 10 years ago at the University of Washington, as Roy mentioned. During my postdoc, I was concerned with the question of, could we design proteins to bind to other proteins? And we targeted the hemagglutinin protein from the flu pandemic that caused the Spanish flu of 1918. So just as a reminder, this pandemic was very, very serious. It caused the deaths of probably 50 or even more, 50 million people or even more around the world around 1918, 1919. So similar to the coronavirus, influenza also has a protein that is responsible for attaching and infecting our cells. This is known as the hemagglutinin protein. And we knew that, again, similar to the coronavirus spike protein, there is a vulnerability on this hemagglutinin protein. We knew that binding tightly to this site can block the functions of this protein and can therefore block infectivity. So we used the methods that I developed in order to design proteins which have this sort of fist-like shape that bind to this region. And this was the first example. In fact, we showed that these proteins bound at atomic accuracy. They bound exactly as we designed them to bind, which was the first demonstration of its sort, that computational protein design could actually target biomedically relevant proteins. And not only were these proteins accurate, they also blocked the infection in multiple flu strains, including the pandemic Spanish flu and epidemic Asian and swine flus, which came later. And actually, they were better blockers than Tamiflu, which is commonly used as a treatment against the flu. So this may sound like a case closed. Maybe now it's possible to use exactly the same method and transfer it to coronavirus spike protein, just generate new binders similar to this in order to target the spike protein. But unfortunately, this is not the case. This method, even though it was quite a breakthrough for computational protein design, the designs that came out of this method were somewhat compromised. They were not optimal. And it took years to optimize these proteins. For instance, they were not stable. They were not high affinity enough. It took years to optimize them to the point that they could actually provide protective protection against influenza virus. And this is time that we simply don't have in a pandemic situation such as this. So over the course of the 8 years or so that I've been at the Weizmann Institute, we've been developing a different computational design strategy in order to address more reliably and more quickly such design challenges. So before I show you some examples that our design strategy actually works in some real world cases, I'd like to give you a little bit of an intuition about what computational design generally means and what is special about our approach. So in computational protein design, we start with an atomic structure, as I showed for the spike protein, for instance, an atomic structure of the protein, and we use a physics-based strategy in order to design proteins to bind to that protein. What do I mean by physics-based? For instance, we represent each atom in the system on the computer. And you can see some atoms would be, for instance, charged negatively. They have an electric negative charge shown in red. Others would have a positive charge shown in blue. So these would attract one another, et cetera, et cetera. So we have essentially, the computer can calculate very, very rapidly the stability of different constellations of atoms in the protein system. So this sounds very nice. In principle, if this is rapid enough, we can go through billions and billions of different solutions and find the best one. But we and others have known for a very long time that such physics-based calculations on their own are not sufficiently accurate to design complex proteins such as antibodies. So the strategy that we developed, which is unique to our lab, uses, in addition to this physics-based information, also information derived from evolution. What do I mean by this? You can think about evolution as a sort of a billion-year general experiment done in nature on countless organisms in different environments. And what we can do is simply look at the surviving structures, the surviving proteins that survived the evolutionary selection process, and ask, what are these structures that survived? And in contrast, what are the structures that were left behind by evolution? They were selected out. And based on that, we can build a model for what structures are beneficial and stable, whereas what the other structures are basically just junk and we should throw them away. So in a sense, our strategy is a hybrid. We use evolutionary data in order to guide the physics-based calculations. So when we started off with this sort of hybrid strategy, I didn't know what to expect. I didn't know whether it would work, and if it does work, to what extent, what would be the scope of this strategy. But what we found out over the past 4 years or so is that this hybrid strategy is actually very, very effective in dealing with very complex problems of protein engineering and design. And I'll show you just two very brief examples for how this hybrid strategy has helped us deal with real-world protein engineering and design problems, starting with the question of stability. So proteins, many proteins, so we should think about proteins as being, the most complex molecules on Earth. And often, many proteins are just simply not very stable. And this is a special problem when you're developing a vaccine, for instance, because vaccines need to be produced in massive amounts. And I'll show you one example for vaccine development from malaria, which we looked at. So malaria is still a very serious disease. It's actually the most serious parasitic disease. It kills 500,000 people every year, mostly young children in developing world countries. And even though there's been over 60 years of research into developing a vaccine for malaria, this is considered a very serious problem, and still there is no vaccine for clinical use. A few years ago, this protein called Rh5 from the parasite that causes the malaria disease was characterized by our collaborators and shown to be a very promising candidate to serve as a vaccine, because this protein is responsible for the process by which the parasite invades our red blood cells. And we know that antibodies that block this protein can actually block malaria infectivity. So this sounds very exciting. Now we have a protein that can serve as a vaccine against malaria, but this protein Rh5, similar to many proteins from pathogens and especially from parasites, is notoriously unstable. It's very difficult to mass produce, and it breaks down at temperatures as low as 40 degrees Celsius. And why are these such terrible problems? Because if this protein ever reaches clinical stage, we would have to produce it in hundreds of millions of doses and ship it to some of the poorest countries in the world, for instance, in Central Africa, where there's no electricity and therefore no refrigeration. So to make this protein Rh5 feasible as a potential malaria vaccine, we must make it much cheaper, and also much more stable in terms of its temperature stability. So we used our hybrid strategy, evolutionary-based atomistic design strategy, in order to design 3 variants for this Rh5 protein, and we recommended these variants to our collaborators at Oxford University. So these funny-looking blots, I'll just give you a little bit of background into biology. These funny-looking blots give you information about the levels of production of the protein in standard bacterial cells. What this shows you here is that the natural protein Rh5 can simply not be produced in standard bacterial cells, which means that the protein is very expensive to mass produce. By contrast, all the 3 designs that we made are massively produced in standard bacterial cells, and already this result brings down the cost of production for this key protein by about an order of magnitude. So this is an important result in itself. Furthermore, this design is also more stable by about 15 degrees Celsius. In fact, it's stable to the point of about 55 degrees Celsius, whereas again, natural Rh5 already breaks down at about 42 degrees Celsius. Our protein is stable beyond 50 degrees. So that means that it can be shipped without refrigeration, even to Africa. The most important question, of course, is it functional? Is it immunologically functional? So our collaborators at Oxford injected natural Rh5 in our design to mice, isolated their antibodies, and tested whether these antibodies provide protective immunity. And as you can see here, the immunological profile for both the design and the natural Rh5 are completely identical, which means that the protein has lost nothing in its biological function. It's still an active, potentially an active vaccine. And this was the first demonstration of its kind for computational design to be able to very quickly, essentially in one shot, address a key biomedical problem of how to mass produce cheaply and economically, and also how to stabilize a key vaccine immunogen for a very important disease such as malaria. So this method is completely automated, and we make it a very important principle for this lab that any automated method should be very easy for other people to use. So we developed this method into a web server, and over the past 3.5 years since launching this web server, you can see that essentially labs all over the world have been using it. More than 1,000 academic labs have been using it daily, actually. Some of them have been using it very often. In the context of the coronavirus pandemic, we see many labs that are using our web servers in order to design better spike proteins, for instance, to use as a potential vaccine. And I'm very hopeful that our servers will actually help in developing a cheap, economical, and fast vaccine for human use in the future. So what I mentioned until now is how do we stabilize therapeutic proteins? Another key question is, can we increase their activity? Now I'll show you some more recent work that we did on a protein that can break down nerve agents. So nerve agents are these very complex chemicals that are also very toxic. They're completely synthetic molecules that are produced by militaries and terrorist organizations, I guess. And one drop on the skin of an adult, or for instance, VX or Soman or Serene, can kill within minutes. An adult can kill an adult within minutes. So these are incredibly toxic compounds, and there's a lot of interest in finding a suitable countermeasure against these very toxic compounds. PTE, this is a natural protein that can break these compounds up, but it does so very, very slowly, too slowly for therapeutic use. So we asked whether the strategy that I mentioned at the beginning, this evolution-guided atomistic design strategy, could help us to design better variants of PTE that would be able to break down these terrible toxic compounds. So we designed a set of these variants of this PTE enzyme, and what this little bit complex plot shows you is the improvement we observe in different enzyme variants relative to the natural enzyme for these toxic compounds, including VX, Serine, and Soman. And you can appreciate this y-axis here is in log scale. So you can appreciate that some of these designs improve the efficiency of this enzyme by from 100 to 4,000-fold relative to the natural enzyme. These two enzymes here actually provide cross-neutralizing protection for all of the nerve agents we tested, including VX, Russian VX, Serene, and Soman. So these are actually at the therapeutic level, and they can be used as therapeutics against these toxic nerve agents. So this, again, provided really unprecedented demonstration that computational design could generate therapeutically relevant enzymes, in this case, therapeutic proteins, in one shot. We obtained these enzymes within 2 or 3 months of starting the project, whereas it took more than a decade for in other studies to optimize proteins to such levels. So very, very quickly to reach therapeutic levels in these designs. And time is critical in these things, because especially in a pandemic situation, because all of the other conventional optimization strategies rely on iterative lab work, which takes months or years to mature. Whereas now we have these, essentially web servers that anybody can access and use in order to optimize their favorite proteins. So now I'd like to go back to the question with which we started this brief talk. Can we design antibodies that would bind to these specific vulnerabilities under a new coronavirus spike protein? So again, this is very, very preliminary. I don't yet have data to show you that we have such antibodies, but I want to share with you the strategy that we're using, which I think is quite promising, the strategy that we're using in order to tackle this problem. So as usual, we start on the computer. We start with the viral protein. In this case, this would be the spike protein. And again, we're not interested in binding to just any surface on the spike protein. There's specific sites, vulnerabilities again, on the spike that we're interested in. And we're using our computational methods in order to dock and to design, essentially to sculpt the surfaces of the antibodies to bind to this surface. This is computationally very intense. It takes really tens of thousands of hours on a computer cluster. But in the end of this process, we get, let's say, a few dozen promising candidates, promising antibodies that have the proper computational metrics, features that encourage us that they may be able to bind this target site. But we don't stop there. The novelty of this method is that we're starting from each of these, we call them leads, and we start from each of these leads and design 100,000 different variants. We're re-sculpting each of these leads to develop 100,000 different variants, mutants of the leads. And in total, we're designing between 1 million and 10 million different antibodies. And we're not keeping this just to the computer. We actually want to test them experimentally. So now we take all of these 1 million to 10 million different antibodies. We have a method to generate all of these antibodies and screen them using an experimental strategy known as high-throughput screening. And this high-throughput screening enables us to test 10 million different designed antibodies essentially within an hour. And we can get data on which of these antibodies bind most tightly to the spike protein, and which of them is better, it just doesn't work. And that can help us to improve our methods and also to obtain high affinity binders. So the coronavirus spike protein is incredibly challenging, but I'm happy to report that we did get a few starting leads that seemed very promising. You can see just examples of them. You can see that they sort of hug the vulnerabilities on the spike protein very tightly, and you're always happy to see such proteins. These are just 2 of the 2 million that we're testing in the lab currently. So you can imagine the scope of this project is quite large. And although we are optimistic about these 2 million designed antibodies, we're also realistic. And we know that this is a very challenging problem. We're applying these design methods now also on a very large scale. We have obtained a donation from various, from a supercomputer in Switzerland and various companies in order to massively produce many, many more such designs. This is a good time to mention the very talented group of people who are working on this project, Die Goldenzweig, Ravit Netzer, Lucas Kraus, and Shai Hoch, who are really working day and night, literally, on this project. I'd like also to thank my group and funding agencies and you for your attention. And just before we go into the questions and answers part of the talk, I'd like to make just one final comment. And I've seen news reports comparing the current coronavirus pandemic to the Spanish flu pandemic with which I started this talk, which caused, again, 50 million deaths worldwide. And that's very, very scary. And although the numbers for coronavirus are indeed quite high, we should be more optimistic than that. That's my sense, at least, because science and technology are obviously in a much better position today than they were in our grandparents' day. We have, what I showed you today could not have been done even 5 years ago. We didn't have these techniques at all. And there are hundreds and thousands of labs throughout the world, each of them with their own new techniques, who are applying their technologies to this problem. So I'm optimistic that a solution would come quite soon. Another reason for optimism, in my view, again, is that we see, since this pandemic started, we see something that resembles, I would say, very rapid, and in my view, a very positive revolution in how science is done, in how science addresses such crises. In usual circumstances, scientists work alone in small teams. They develop their methods, they get data, they gather these data, and then they send these data in publication to a specific journal. They wait for the journal to respond, and this process can take months and even years in some cases. What we're seeing with the corona pandemic is groups publishing even raw data on web servers, just for other people, for other groups, to very rapidly build on these results and thereby accelerate research. And we are seeing just very, very rapid response to this coronavirus. And I think this is incredibly, it's both inspiring and also promising with respect to this coronavirus pandemic. And maybe this is a good time to also think, more broadly and for the long term, that it is really these sorts of collaborations that can solve massive problems of such global scale. And this pandemic is very serious, but it's not the last pandemic that humanity will face, and it's not the most serious problem that we'll face in the future. There's still the climate change. There's other societal and economic challenges that we need to face, and if we face them together through collaborative technological, scientific, and governmental efforts, I think we have a much better chance of favorably solving them. So with that, I'll close and I'll answer any questions to the best of my ability.
So the first one is, I've read that COVID-19 has mutated into many variants. Is it possible or even likely that the virus in New York City and Italy is a different variant than the one on the West Coast, say in Los Angeles?
This is a really good question. I think it's still an open question. I mean, it's very clear that the virus is mutating. It's an open question whether these mutations are providing an adaptive benefit, whether these variants are better adapted to infect us or not. It's not clear at this point. So far, the scientists I've talked to think that this is not the case, that the mutations we observe in coronavirus are not substantial for its infectivity. It doesn't mean anything for going into the future. And I should mention in this context, influenza. So in the case of the flu, we had this massive pandemic, the Spanish flu pandemic of 1918. After a huge amount of the population, a huge percentage of the human population became immune to the Spanish flu pandemic, this virus, the flu, acquired mutations. And these mutations enabled it to continue to infect human populations. But these mutations also debilitated the virus. It became from this massive, very aggressive pandemic into what we now know as seasonal flu, a much less serious disease. So these mutations, they may become prevalent in the viral populations, but they may also weaken the virus in the future. So, it goes both ways. We shouldn't be so afraid of these mutational processes, actually.
Great. I'll move on to the next question. Are the antibodies specific to a person or specific to a type of virus? So, can we be sure that an antibody derived from one person and used in another will work the same way in the second person as it did in the first person?
A very good question. In general, the answer is yes. The antibodies would be specific for the virus and would behave nearly the same across the population. Why I say nearly the same? Because some antibodies can cause an immune reaction, so our bodies may treat them as foreign and therefore react to them. But this would take time. It doesn't happen overnight. It actually may take months to happen. So in treating an acute disease such as a viral infection, this doesn't become a serious issue. But it's an important point. This is one of the reasons why drug development is such a lengthy process, because even when we find even a human antibody, which is considered to be state-of-the-art, gold standard safe, we still need to make sure that it is indeed safe in a large cohort of humans.
That's excellent. So I'll do some follow-up questions, which I think are perfectly suited to where you ended with that. Does the introduction of synthetic antibodies interfere with the generation of naturally produced antibodies after the initial synthetic antibody treatment has been completed?
Not that I know at all. The antibodies would essentially exist in the system for as long as they're stable. And this can be from a few hours to a few days, sometimes weeks. They're not there to stay. So in one very attractive feature about antibodies in general, some antibodies may behave differently, but in general, antibodies are incredibly specific. They would bind to the desired target and usually not to anything else. And again, there are safety concerns always. When you inject a massive amount of protein to a person, you first need to test it in animals and in a small group of humans. You don't go and deploy on a massive scale before making sure that it's safe. So in general, I would say the answer to this question is no, it's going to be fine. It wouldn't interfere with the natural process of our immune reactions. But you have to test these things.
So this is, again, related to the synthetic antibodies. Given the neurological impact reported as a consequence of this virus, can these synthetic antibodies cross the blood-brain barrier to neutralize the virus directly in the central nervous system?
Very, very knowledgeable question. Yes, so there are some reports on the virus actually infecting the central nervous system, which is very worrying. Usually the disease happens within the lungs more typically, and that means that the usual course of action with such an antibody is actually with an inhaler. You would inhale an aerosol containing such antibodies or other drug molecules, and they would protect your lungs. It seems like, I'm not an expert in this, but it seems like these central nervous system infections are due to very severe disease. So hopefully if we can get a hold of the disease in an earlier state, we wouldn't need to cross the blood-brain barrier. Typically, antibodies don't cross the blood-brain barrier. There are some modifications that may enable that, but generally speaking, this is not the case. They typically don't cross the blood-brain barrier.
That's great. I think one question that's more clinically related, but I can ask you anyways. Somebody's asking if there's anything that we can ingest or any nutrient that we can have that actually helps boost the human immunity response in creating more, greater quantities of antibodies.
Not that I know.
Okay, thank you. Another question related to antibodies. Is the size of the dose of the antibody used in a person critical to eradicating and attacking the virus entirely? And then as a related question is not just the size of the dose, is the timing critical for the application of the treatment?
Right. This is very good questions, both of them. The dose is critical because if the viral load is large, you need a lot of antibody, of course. There's also some questions about the kinetics of blocking by antibodies. These are largely questions that we cannot address theoretically. They need to be done experimentally. And this is, again, one of the reasons why it takes time to develop a drug. Even having a very promising candidate in the lab, we still need to know what dose to use in a real world situation. The second question was about, I'm sorry, this was?
It was related to the time, so the timing.
Yes, that's also a very, very good question. Some drugs can be given as a prophylactic. Meaning, imagine nurses and doctors working in a highly contaminated area. You want to protect them as much as possible. If they could take an inhaler, for instance, to protect their lungs as they're treating patients, that would be absolutely fantastic. So there's a question of what is the therapeutic window? And again, you cannot address these questions theoretically, you have to test. It would be ideal if you could provide such a drug prophylactically. And then the question is, after infection, how many days post-infection can you still get the therapeutic response? Again, this is not a question for theory. It's something that we need to address experimentally by actual testing. Very good questions.
This is related to that note of optimism that you spoke about in the scientific community really coming together at a moment of crisis. Do you think that this more open kind of collaboration will continue, or do you think that there'll be a reversion after the crisis is passed back to the lone scientist in competition model?
Again, a really great question. I'm hopeful. I'll explain. Maybe we shouldn't be too critical of how science is typically done. The reason why scientists take so much time to publish their work is because they're careful. We want our scientific papers or work to be, not here for just a few months and then be shown to be wrong. We want them to have staying power for months or years, or hopefully even longer than that. So typically scientists are careful because they want their scientific results to be robust. So I think when we end this emergency, I think a lot of science will go back to being done more carefully over longer periods of time, and that's just perfectly fine. I think, though, that we need to think about how do we address future crises as scientists and as technologists. And in that case, we want to break down barriers as quickly as possible. And we want to make sure that there are these forums for exchanging ideas, talking to one another as quickly as possible in times of crisis. I think it's more a solution to this than to a general, science as a whole.
So another question, does the spike protein mutate? And if it does, would the antibodies that are being designed still be effective?
Excellent. Yes. So this was really the first question on this talk, was how the virus mutates. And in fact, what we know is that mutations are happening on the spike protein. And yes, this is key, because what this means is that antibodies that target regions that are mutated have a very high chance of simply not working in the future. The sites that I showed you as vulnerabilities are actually highly conserved. What does that mean? Why are they vulnerabilities? The virus absolutely needs them in order to infect our lung cells. And that means that these sites are conserved, not just in this coronavirus, but also in the Sars virus, the virus that caused the Sars epidemic of 2003. And this is the reason why we're targeting these sites specifically, because we think, there's no assurance in these things, but we think that the virus will have a hard time mutating these sites without losing its ability to infect us.
So one question that somebody sent in is, how do we account for the virus presenting totally different symptoms in different individuals? Some people, it can be completely asymptomatic, and other people can, develop, life-threatening pneumonia from it. Any kind of insight into that?
It's an open question. I think many people are studying this. There are some, I'm not an expert, but what I've heard, for instance, is that there's obviously a demographic issue here. Older people get more seriously ill, typically. One of the reasons that I heard about this is that the lungs may have become weaker with age, but we don't yet have an answer. This is, we have had this virus for only a few months. There are still no good animal models to study this virus in the lab. People are working actually very hard on developing animal models for this. Many of these questions really need long-term research, and this research is being done right now. These are excellent questions. We need to know the answers to these questions.
I like this one somebody sent in. Is folding at home still a useful tool for developing molecules for your effort? You might want to explain to people what that is.
Yes, so there's folding at home, and there's actually Rosetta at home, which is a little bit more relevant to this question. These are what's called peer-to-peer servers. People are donating their computer resources. You can donate your computer at home, your Xbox, or whatever you have to these servers. And when you're not using the computer gets information from a centrally located server on how to run some simulations on proteins. Some of these simulations have to do with actually designing proteins. We're not using these servers, again, as I mentioned a little bit before, we're using a supercomputer that was donated to us by a very large Swiss group at EPFL. We're also using Amazon Web Services, which were very generous in providing us access to their computational resources. This again shows you this sort of generosity that we see, not just in science, but also in technology. People are really, they've heard about our research, and they've approached us to see if they can help. And this is, it's again, inspiring.
Somebody asked a question about a product, a word I think none of us would have ever heard of if it wasn't for this, which is remdesivir.
Yeah.
Your thoughts on it as a treatment and its mechanism of action was what they were asking about.
Yeah, it's really good. I don't know enough. This drug I can't even pronounce. Its name properly. Is a general antiviral, and it's being tested right now. I think taking off-the-shelf drugs has a huge potential, but we have to treat these very carefully. Chloroquine became very prominent in the news a few weeks ago, but it was very clear from the start that the therapeutic window for that was iffy at best, and now some results came from Brazil showing that it was a complete mess, and that people should definitely not use it. So again, each one of these drugs must be studied very carefully. I know that there's a lot of work in China right now. There's also amazing work being done in Italy. Doctors are really literally pulling stuff off the shelf and trying to treat their very difficult and challenging patients. And I've already seen, again, slides. Again, people are not waiting to have data for papers. They're just sharing slides of data from hospitals, and it's just amazing to see. So yeah, another reason for optimism is that we do have our shelves are highly stocked.
So I'll make this the last question, because there are a lot of others, but if you were going to try to predict, what is the probability that in a secondary wave of COVID infection, the virus will present differently? Yes.
Okay. So I think it's an open question, and I think making predictions on exponential processes is very, I won't go that way. But one thing that I can say, just as a comparison to influenza, which again serves us as a sort of a reference in this very serious situation. Influenza is known to undergo relatively fast mutation, but there are reasons why influenza is more mutagenic than the current coronavirus. I won't go into the details of this. It's a little bit technical about how the viral genome is organized. But the viral genome is organized in influenza in such a way that enables it to undergo very fast mutation, whereas in coronavirus, this is not the case. And that gives, again, the experts reason for optimism that we won't see, we may not see something like seasonal coronavirus. Maybe this won't be as serious as how influenza has been manifesting itself in human populations. But, again, I'm not going to make a prediction on this.
Great. So I want to thank everyone for participating today and really great questions. On behalf of all of us who participated, Sarel, Roe, thank you guys. I want to, on behalf of all of us, thank all the scientists, all of your students that are doing such amazing work combating this global epidemic at the Weizmann Institute. I just wanted to say that I find myself recharged and feeling a little bit more optimistic and hopeful after each one of these webinars. So thank you so much for giving us a glimmer of hope here. And on behalf of the American Committee network of supporters and donors, we're really proud to be able to help you guys get the resources and that you need in order to pursue these really interesting scientific possibilities. To that end, I just want to remind everyone that we've announced a global fundraising drive to reach $25 million for research related to the novel coronavirus that's happening right now, and we're raising money that really throughout the world. I want to thank many of you who are on this call for generously supporting that effort and contributing. I'm proud to announce that we've already raised millions of dollars since the beginning of the initiative. And for those of you who are still considering or would like to get involved, I encourage all of you to either reach out to your local Aquis professional, or you can just send me an email. I hope all of you are safe and healthy and look forward to the next time we can all physically be together.